So while I get the presentation where the last one was right after lunch, so everybody was digesting, now's when everybody has to go to the bathroom, right? So hold it for just a little bit longer. So I'm Tim. I'm the president and CEO of Abeona Therapeutics, and I'm here to tell you a little bit about our company today. So we are a publicly traded company on the NASDAQ exchange. So of course, here's our forward-looking statements slide. So we have a number of, we're a gene therapy company or a rare disease company. We have a number of clinical programs, 3, and a number of pipeline products that are using some of the same products or the same technologies. So today we're going to talk a little bit about our lead product for MPS IIIA, which is a lysosomal storage disease. Similarly, we also have an open IND for MPS IIIB and for our Epidermolysis Bullosa program, which was just published yesterday in JAMA. We have 2 upcoming preclinical programs that are expected to be in clinical trials. These are AAV-based gene therapy programs, also for the treatment of lysosomal storage diseases, for juvenile Batten disease and for infantile Batten disease. And we've announced a few weeks ago the in-licensing of a 60-vector capsid library for the next generation vectors, which I think will be discussed in the panel discussion coming up very soon after this. Stay tuned. So we also have announced recently that we received Fast Track designation status, and if you're following along in the rare disease gene therapy space for companies like Avexis and of course in the post-Sarepta world, these things do have value as you're trying to go through the regulatory filings. So again, just kind of diagrammatically looking at our pipeline, we have 4 metabolic programs. These are AAV gene therapy programs. Our fun little check boxes over here, check back early, check back often. There's going to be lots of additional ones as the year goes on, as we've continued to apply for many orphan product designations both in the US and in the EU. And our lead program has orphan product designations in the US, EU, the rare pediatric disease designation, which is on track to the priority review voucher, and a fast track, which does allow us to file a rolling BLA. We have a dermatology program in Epidermolysis Bullosa, which is in phase 2, and some hematology programs and plasma therapies, which I won't be talking about today, but happy to take questions on it afterward. So we wouldn't be able to do all of what we've done so far without help from the investment community, but also from our global support from foundations. Part of the history of Abeona, which is named after the Roman goddess who is protecting children as they leave home. These foundations all had a mandate to find and fund therapies that they thought were going to be the best hopes for their kids. And they got behind Abeona and invested in a great venture philanthropy model. So it's a great story about how the foundations have seen a return on their initial funding as well, once we went public. So our clinical stage programs to talk about Sanfilippo syndrome type A and type B, or ABEO 102 and 101. One of the things that we note when looking at gene therapy programs, gene therapy is all about delivery. You hear this word delivery, but it really does make a difference. And AAVs come in many different numbers, so AAV 1, 2, 9, 10. Our programs use the AAV9, which is the only vector that's able to cross the blood-brain barrier. So for diseases where you have a profound central nervous system manifestation, you need to be able to get access to the CNS. Now you can do that by drilling into the skull or going into the spine. We've chosen to go for the one-shot cures-all approach, which is using the AAV9 by a systemic administration. So we give AAV9 systemically to treat the whole body manifestations of these diseases. Importantly, what you can see here is just that the AAV9 has a tropism much better than 10 or 5, for example, for central nervous system tissue. This is direct injection into the brain. The references are right there. And importantly, when you look for months to years after injection, again, these are animals that were injected at about 4 weeks of age, looking at 5 months or 1 year post. Basically, what you can take away here is more green equals more gene, and that's what you want when you're trying to do with these slides. I heard somebody laugh over there. But really what you're showing here is that the viral vector was able to get access into the CNS and basically broad and robustly distribute throughout the CNS for expression. And now in animal models, and now we're starting to see clinical data in Avexis and our program and others, where you're really starting to see clinical demonstration of not just proof of concept, but early biopotency leading to efficacy. So our programs are in Sanfilippo syndrome. So these are lysosomal storage diseases. 70% of these kids don't reach the age 18. They're universally lethal. The kids are often diagnosed between the ages of 2 and, say, 5, and they progress to a point or develop to a point and then spend the rest of their life declining. So to prepare for clinical trials, and you heard this story many times repeated in the press over the past year, is really we did a natural history study, and to highlight the importance of natural history studies is to validate these outcome measures. So we had 25 patients go through 1 year of follow-up, and we were able to assess neurocognitive function, some motor tests, looking at standard serum and clinical chemistries. We did MRIs to be able to assess organ function and size. But again, all of these are through 1 year, and multiple patients are crossing over now into the clinical trials, so we have long-term longitudinal data to be able to compare against. And really what the natural history study allowed us to do was, if you look at all of these patients, their follow-ups, they are all declining. Okay, and there's a floor effect that you can see here, and this is a normal range of children up above this in this gray area. But again, what this helped us do was validate a nonverbal IQ score and a behavior assessment as neurocognitive functions for us to be able to assess as potential ways to look at efficacy going into the clinical trials. And importantly, we also did MRI data looking at the livers and spleen. Now, these kids present with enlarged livers and spleens, and what we saw in the natural history study was that they came in at baseline and they had about 100% enlarged spleen and livers that when they came back a year later had tracked with them. The kids had grown bigger, but so the livers and spleens had as well. So they saw that continued part of the disease manifestation. And so as we got into the clinical trials, and this is our general clinical trial design, looking at an open-label dose escalation, low dose and a high dose. We've completed enrollment in the low dose, and we've recently announced that data. And enrollment in the high dose has commenced. Primary objective is safety with initial determinations of potential efficacy as measured by reductions in urine, and CSF gag, disease manifestation in the liver and spleen, increased enzyme activity levels, and then all these neurocognitive assessments. And the fun stuff is, looking at this data. So here we see a reduction of about 25% gags. So the gags are the sugars, okay, that the kids can't break down in the lysosomes. And what we're able to show here is that this is proof of concept that the virus, after an intravenous injection, was able to cross into the blood-brain barrier and reduce the holy grail here, was reduce the sugars in the CSF. And then also looking at the urine. So this is a 60% reduction of GAG content over baseline in the urine. And we start, at this point, what we end up doing, just many of you in the audience, is trying to compare that and say, okay, well, what are some of the other products that have come out on the market or are going to come up for commercialization soon? So looking at Ultragenyx, for example, and their MPS 7 program, seeing again a reduction of 65 or 70% GAG in the urine, in this case, dermatan sulfate. And then looking at liver and spleen volumes, so recall 30 seconds ago, I think it was, I talked about the liver and spleen volumes that these kids have a much larger spleen and liver. We were able to see a reduction of about 20% of both liver and the spleen after 30 days. And though not commented here, we also saw increased enzyme activity in the plasma. So taking together increased plasma enzyme activity, reduction of CSF and urinary gags, and a disease manifestation change as in a reduction of liver and spleen. This is what we believe to be best in class at this point. Nobody else has been able to demonstrate this for these types of lysosomal storage diseases, all of these things. And again, just looking again at something like Cerezyme from Genzyme, for Gaucher, again, you're looking at, these are things that regulatory agencies look at when trying to consider up for approval as registratable endpoints. So, with that, these are exciting programs. We're looking forward to announcing our first patient in the high dose coming up soon. Stay tuned. We also have, for a very similar program in MPS IIIB, this is the preclinical data showing that we completely corrected the GAG content as well as the survival deficits in these animals. We have an open IND and look forward to enrolling that patient in the upcoming months. And now to switch gears just a little bit to probably the most horrible disease you've never heard of is Epidermolysis Bullosa. It's a skin disorder where these kids, if they bump their arm, it's also known as butterfly skin, the skin blisters and falls off. These kids can have wounds up to 40 to 60% of their body. They spend their whole lives in bandages. They lose their fingers and toes, and they're in pain pretty much all the time, and they're wrapped in bandages. They can spend hundreds of thousands of dollars a year just in bandages alone. There are no therapies for them. What we do is we do an ex vivo correction of autologous cells. We take a punch biopsy, grow the cells up, correct them using a retrovirus, grow them into these sheets over here, and then graft these on. And each child receives about 6 grafts, and each graft is about the size of an iPhone. So just to help put that in perspective, everybody's got a phone, so now you know how big the grafts are, right? So what we saw here, and this was just published in JAMA, this is about the size of a wound, and we put these grafts on, and the wounds stay healed for some of them up to 12 months. So when you consider that the standard of care for these is something like Dermagraft or Apligraft, those fall off in about a month. So again, what we're trying to do is help these kids heal their wounds. As a biomarker, we're able to take a punch biopsy at the edge of the graft and show that we are able to recapitulate the actual underlying deficit, which is the COL7 collagen expression. And you can see here the immunofluorescence showing that we've restored the actual collagen expression. And so now we've gone through 5 patients in the phase 1. We are now enrolling in phase 2 clinical trial at Stanford University. And again, we're looking to enroll about 5 or 6 more adolescent patients. And just to touch base on our preclinical programs, we have juvenile Batten disease, infantile Batten disease, and a program in gene editing for Fanconi anemia. One of the great things about these programs, particularly in the metabolic space, is that they use the same AAV, pretty much delivered the same way, and all of the preclinical modeling is consistent across all programs, across all the sites. So preclinically, we're seeing complete correction of survival deficits, neurocognitive function, and neuromuscular function. So now getting into clinical trials and seeing some of the early data in our Sanfilippo syndrome type A has been very, very gratifying, because we believe that that's going to translate through the rest of our programs. And importantly, this is where we just got off of a fundraise, and we just announced that we did a follow-on public offering. But we're just like a Vexus, like a year ago. They're using the same vector, delivered the same way, for a very similar type of disease. So human proof of concept is continuing to be developed for these AAV gene therapy programs, as well as their safety. And 1 or 2 more slides on the infantile Batten's disease as well. Again, this is another lysosomal storage disease. Affects the kids. It's lethal, often by the age of 5. And again, the therapies have corrected the survival deficit as well as some of the neuromuscular functions. And I'll close with our in-licensing strategy. Many of you know that when you give these AAV gene therapies, you develop an immune response. And these therapies, while they may last for potentially a decade, at some point these kids may need another dose of something else. So our strategy has been to go after a vector library. Some of these have equal or better CNS tropisms to the current standard ones, and so we look forward to reporting more data on that in the future. And with that, here's our board, and I'll close with contact information. Thank you.